In 1977, a submersible named Alvin descended into darkness near the Galapagos Rift. Nearly 2,500 meters down, the ocean should have been quiet. No sunlight, near freezing water, pressure more than 250 times greater than at the surface. The crew expected volcanic rock and nothing more. Instead, their lights caught something rising from the seafloor, black towers, columns of superheated water blasting into the abyss. Instruments recorded temperatures above 400 degrees Celsius, hot enough to melt lead. At the surface, water would explode into steam at those temperatures. Down here, under crushing pressure, it remained liquid, and wrapped around those vents, clinging just centimeters from water that could flash boil at sea level, were living creatures. Tall white tubes crowned with red plumes, crabs, mussels, shrimp, not scattered, dense, thriving. There was no sunlight, no photosynthesis, no plants forming the base of the food chain. By every biological assumption of the time, complex life required sunlight, and yet surrounding these vents were dense communities of large animals positioned just centimeters from fluid, hot enough to melt lead at the surface. Proteins normally lose their structure at extreme temperatures. Cell membranes become unstable. DNA can degrade under chemical stress. Vent fluids are acidic. They carry dissolved metals. They emerge from the crust at temperatures exceeding 400 degrees Celsius. And still, life organizes itself here. So what exactly is happening at these vents? What allows life to anchor itself beside something that should destroy it? And how far can biology stretch before the word impossible loses meaning? Tonight, we examine the deep-sea creatures that live in boiling seawater, a boundary that rewrote biology. But before we begin, if this space brings you peace, a like or subscribe helps other curious dreamers find their way here too. And I'd love to know, where are you listening from tonight? What time is it where you are? It's always lovely to see how far this calm corner travels. So now take a deep breath, let your shoulders loosen, and together let us descend toward the vent fields where heat becomes the foundation of life. At the surface of the sea, water follows familiar rules. Warm it to 100 degrees Celsius, and it trembles, then erupts into vapor. Bubbles form where liquid turns to gas, rising and bursting into the air. Boiling is simply a rebellion against pressure. The surrounding air presses lightly on the surface, and when heat gives water molecules enough energy to push back, they escape as steam. Far below, that quiet balance changes. 2,000 to 3,000 meters down, the ocean bears down with immense weight. Every square centimeter carries the force of hundreds of atmospheres. Under such compression, water molecules are held in a tight embrace. To break free into vapor now requires far more energy. The boiling point climbs steadily as pressure increases, so high that temperatures which would create violent steam at the surface leave deep water strangely composed. Near hydrothermal vents, this principle shapes an otherworldly stage. Seawater seeps into cracks in the crust, sinks toward hot rock, and absorbs heat from magma beneath. As it warms, it does not boil. Instead, it becomes superheated, reaching temperatures above 400 degrees Celsius while remaining liquid. The crushing pressure prevents explosive expansion. The fluid grows less dense, enriched with dissolved minerals and metals, and eventually surges back upward through chimney openings. What emerges is not a rolling boil, but a dark plume, shimmering and turbulent. The fluid mixes instantly with surrounding seawater that hovers near 2 degrees Celsius. Within centimeters, temperature can plunge by hundreds of degrees. The boundary between extremes is sharp yet unstable, folding and curling like ink dropped into cold glass. Here, physics paints gradients rather than uniform heat. Boiling underwater rarely resembles the vigorous bubbling of a kitchen pot. Gas does not freely expand into open air. Instead, dissolved gases remain trapped until pressure lessens. When vent fluid exits into slightly lower pressure water, tiny mineral particles precipitate, forming smoky clouds. The drama is chemical and thermal rather than explosive. Heat disperses through mixing, conduction, and convection, each process shaped by density differences and pressure constraints. Instruments lowered into these plumes reveal a layered landscape. A probe can move a few centimeters and record a shift from near freezing to scalding. These gradients create narrow corridors of possibility. No creature anchors itself within the hottest jet. Instead, life occupies the margins, where temperatures fluctuate within survivable bands. The vent chimney becomes a scaffold around which microhabitats arrange themselves, each defined by a delicate balance between heat and cold, chemistry and dilution. Superheated water remains liquid because pressure alters the phase diagram of water itself. The boundary between liquid and vapor bends upward with depth. Only when fluid rises and pressure drops does it approach the point where vapor can form. At the seafloor, however, the ocean's weight holds molecules close, preventing the violent separation that would otherwise occur. The result is a paradoxical environment where water exceeds surface boiling temperatures, yet flows like any other current. Within the rapid mixing zone, survival depends on precision. Too close to the vent orifice, and proteins unravel. Too far away, and chemical energy becomes scarce. The steep gradient allows organisms to position themselves within centimeters of stability. Their existence is shaped not by enduring 400 degrees, but by inhabiting fleeting intersections where heat has softened and cold has not yet erased chemical richness. Thus, the stage is set by pressure and temperature, by physics that bends familiar rules without breaking them. In this compressed darkness, water refuses to boil, and in that refusal, space is carved for life to gather along the edges of fire. They rise from the seafloor like pale spires, taller than a person, slender and still, each crowned with a crimson plume that drifts in the faint currents. More than 2 meters in length, these beings stand anchored in mineral crust, their bodies encased in smooth white tubes. They do not hunt, they do not graze, they do not even possess the simplest opening through which food might pass. Riftia pachyptala lives without a mouth, without a stomach, without any digestive tract at all. Inside the tube, its soft body is divided into regions, each specialized, each precise. At the top, the red plume extends outward, richly supplied with blood vessels. It resembles a delicate feather, yet it functions as a living interface between animal and ocean. Oxygen from the surrounding water diffuses inward. So does hydrogen sulfide, a compound toxic to most known creatures. Both substances travel through an intricate circulatory system filled with an unusual hemoglobin. This molecule binds oxygen as hemoglobin does in many animals, yet it also binds hydrogen sulfide without being poisoned by it. The two chemicals are carried together through the worm's body, not to a stomach, but to a dense internal organ known as the trophosome. The trophosome occupies much of the worm's interior, a soft brown mass composed not of worm tissue alone, but of billions of symbiotic bacteria. These microbes are the true chemists of this partnership. Using oxygen as an electron acceptor and hydrogen sulfide as an energy source, they perform chemosynthesis, producing organic molecules from carbon dioxide dissolved in seawater. In place of digestion, there is chemistry. In place of feeding, there is exchange. The worm provides transport and protection, positioning its plume where chemical gradients are favorable. The bacteria provide nourishment, converting inorganic compounds into sugars and other carbon-rich molecules that diffuse into the worm's tissues. Host and microbe are bound so completely that neither survives long without the other. The worm's body plan has evolved around this relationship, relinquishing structures that once defined animal life. Growth occurs with remarkable speed. In an environment where energy flows steadily from the crust below, Riftia can reach full length in less than 2 years. Its tube lengthens upward as the animal extends, secreted layer by layer from a specialized gland. The plume expands, increasing surface area for chemical uptake. Within a short span of time, a larva scarcely visible becomes a towering presence among vent communities. The larval stage begins differently. Young worms possess a simple gut, a temporary structure that allows them to ingest bacteria from the water column. Once symbionts establish themselves within developing tissues, the digestive tract degenerates, the trophosome enlarges, and the architecture of dependence is sealed. An animal relinquishes autonomy in exchange for metabolic certainty. When scientists first examined these worms, the absence of a mouth seemed almost implausible. Complex multicellular life had long been understood through the lens of ingestion, of food webs beginning with plants and passing through herbivores and predators. Here was an organism that bypassed that familiar chain. Its survival rested not on consuming other life, but on hosting it internally. The discovery suggested that animal form could be reshaped by environment more profoundly than expected. Organs once considered essential proved negotiable. Physiology adapted not merely to tolerate extremes, but to integrate them into its very design. Within the quiet chambers of the trophosome, chemistry replaces chewing and partnership replaces predation. Along the margins of heated rock, a creature stands tall without ever taking a single bite, sustained by invisible labor within its own living core. Along the rough flanks of active chimneys, where mineral walls shimmer with rising heat, a different kind of worm presses its body against stone. It does not tower upward. It stays low, threaded through narrow crevices, its gray form edged with delicate filaments. This is the Pompeii worm, Alvinocaris pompigiana, an animal that settles closer to venting fluid than almost any other known multicellular life. Its back is often described as flecked with pale fuzz, a dense covering of filamentous bacteria that colonize the worm's surface. Under magnification, these microbes form a living mantle. They cluster in thick mats, especially along the dorsal region that faces the warmest flow. The worm positions its head toward cooler water, while its tail remains nearer the heated rock, creating a body stretched across a steep thermal gradient. Temperatures at one end may be near 2 degrees Celsius, while the other end can endure bursts that exceed 50 or even 80 degrees in localized pulses. These shifts are not steady. Vent chimneys breathe in irregular rhythms. Plumes surge and recede. Small fractures redirect flow. The Pompeii worm experiences sudden fluctuations, brief spikes, followed by cooling, a constant oscillation between extremes. Its tissues must tolerate rapid change without losing structural integrity. Proteins, those folded chains that enable metabolism and movement, normally unravel when heat disrupts the bonds holding them in shape. Once unfolded, many cannot refold correctly. In most animals, such damage cascades quickly through cells. Yet this worm persists in conditions that would destabilize familiar biology. Laboratory studies have examined enzymes extracted from vent organisms, revealing increased proportions of amino acids that strengthen internal bonding. Heat-stable proteins often contain more ionic interactions and tighter hydrophobic cores, features that resist thermal agitation. Cell membranes also adapt. The lipid molecules forming their bilayers shift in composition, incorporating saturated fatty acids that remain cohesive at elevated temperatures. Stability becomes a matter of chemistry tuned over generations. the bacterial coat may add another layer of resilience. Some researchers propose that these surface microbes consume part of the chemical flux emerging from the chimney, altering the microenvironment immediately around the worm's body. Others suggest the bacteria form a protective buffer, absorbing thermal energy or detoxifying compounds before they reach underlying tissues. The relationship appears intimate. The worm secretes mucus that feeds the bacteria, and in return gains a modified boundary between itself and the vent. Experiments using pressurized aquaria have attempted to recreate aspects of this habitat. Specimens brought to the surface without pressure control rarely survive, making study difficult. Under controlled conditions, short-term exposures to high temperatures reveal tolerance beyond that of most marine invertebrates. However, sustained exposure to extreme heat remains lethal. The worm's survival depends on transient pulses rather than constant extremes. It occupies a shifting line where maximum temperatures are encountered briefly, not continuously. Such findings refine the idea of heat tolerance. It is not a simple number marking a limit, but a dynamic interplay between duration and intensity. A cell may withstand a high temperature for seconds, yet fail if held there for minutes. The Pompeii worm lives within that narrow window, its body oriented to balance cooling currents and heated surges. Behavioral positioning complements biochemical adaptation. Pressed against mineral walls that radiate warmth from within the earth, the worm does not seek the hottest jet, but the fluctuating margin beside it. There, among mineral grains and microbial films, it endures conditions that redefine the upper bounds of animal life, holding its place where chemistry and heat meet in constant motion. Light never arrives here, not even as a memory. Beyond the reach of sunrise and moonrise, vision loses its usual meaning. Yet, movement continues across the mineral landscape, delicate limbs stepping through drifting particles, antennae sweeping in slow arcs. Among these inhabitants are vent shrimp, pale and translucent, their bodies shaped by a world where brightness holds no advantage. Many species possess eyes that are reduced or transformed. In some, the familiar spherical structure is absent altogether. In others, the eye persists only as a flattened plate beneath the cuticle, lacking the complex lenses that focus images in shallow waters. These modified organs do not form pictures. They register gradients, subtle changes in radiation, shifts in the environment that replace sight with sensation. One species, Rimicaris exoculata, gathers in dense swarms near active chimneys along the mid-Atlantic ridge. Its back bears a dorsal organ, sometimes described as an eye, yet it functions differently from those of surface crustaceans. Within this structure lie photoreceptor cells tuned not to visible light, but to faint infrared glow emitted by hot surfaces. The vent does not shine in colors familiar to human perception, yet it radiates energy. The shrimp sense that warmth as orientation, aligning their bodies along gradients invisible to us. Heat becomes a compass. Moving too far from the source means losing access to the chemical flux that sustains the microbial communities coating their mouthparts and gill chambers. Approaching too closely risks exposure to damaging temperatures. The shrimp hover within a narrow band, adjusting position with minute flicks of their swimmerets. Their antennae detect chemical traces in the water, while their modified visual organs register thermal contrast. Together, these senses replace the need for image-forming sight. In total darkness, pigmentation fades. Many vent shrimp appear ghost-like, lacking the deep reds or blues common in other deep-sea animals. Color offers no camouflage when no light penetrates. Structural adaptations take precedence. Exoskeletons thicken to withstand pressure. Gill surfaces enlarge, maximizing exchange with surrounding water. Some species harbor symbiotic bacteria within specialized chambers, allowing them to derive nutrition from chemicals dissolved in the flow. The absence of light also reshapes behavior. Courtship displays based on visual cues become unnecessary. Instead, chemical signals guide interactions. Pheromones disperse through currents, carrying information about readiness and species identity. Tactile contact confirms recognition. Eggs are brooded beneath the abdomen, sheltered until larvae emerge into the surrounding water column, where they drift before settling near suitable habitat. Neural architecture reflects these priorities. Brain regions dedicated to processing complex images are reduced, while those associated with chemosensation and mechanoreception are emphasized. The shrimp interpret the world through gradients of temperature and chemistry, through pressure waves and subtle vibrations transmitted through water. Darkness does not imply emptiness, only a different arrangement of perception. Infrared detection in these animals remains an area of ongoing study. The wavelengths emitted by vent surfaces are longer than those perceived by most marine species. Specialized photopigments within their dorsal organs may respond to this radiation, converting thermal energy into neural signals. The precision is not high-resolution vision but coarse awareness, sufficient to distinguish warmer rock from cooler surroundings. Within this realm, sight, as commonly understood, has been relinquished, replaced by senses tuned to heat and chemistry. Movement continues without beams or shadows, guided by signals woven into the very fabric of the environment. In a place where fire glows beyond visible color, perception adapts, and life proceeds without ever needing to see. Energy is the quiet currency beneath every living system. In forests, it falls from the sky as light. In grasslands, it filters through leaves. In shallow seas, it glimmers across drifting algae. For centuries, biology traced nearly every food chain back to that radiant origin. Green cells captured photons, built sugars from carbon dioxide and water, and formed the base upon which all other creatures fed. The pattern seemed universal, a rule as steady as gravity. Here, that rule loosens. At hydrothermal vents, sunlight never enters the equation. Instead, the foundation rests on chemistry rising from within the planet. Seawater circulates through fractured crust, reacts with heated rock, and returns enriched with dissolved compounds. Among them is hydrogen sulfide, a molecule carrying chemical potential, energy stored in its bonds. To most animals, it is toxic, binding to respiratory enzymes and halting metabolism. To certain bacteria, it is opportunity. These microbes perform chemosynthesis, a process parallel in purpose to photosynthesis, yet driven by different fuel. They oxidize hydrogen sulfide, transferring electrons through cellular pathways that generate usable energy. That energy powers the fixation of carbon dioxide into organic molecules. Sugars and other carbon compounds emerge not from captured light, but from chemical gradients. The transformation is quiet and continuous, occurring within microscopic cells that coat mineral surfaces or live inside animal tissues. In this system, bacteria occupy the role plants fill at the surface. They are primary producers, converting inorganic substances into biomass. Upon this base rises a layered community. Some organisms graze directly on bacterial mats, scraping surfaces with specialized mouthparts. Others, like the tall tube dwellers described earlier, host symbiotic bacteria within their bodies, internalizing the process of production. Predators feed on grazers, scavengers consume remains, and decomposers recycle material back into dissolved form. The energy pyramid at a vent is narrower than that of a forest, yet it follows familiar ecological geometry. At the broad base lie countless microbial cells, dividing rapidly where chemical flux is steady. Above them stand invertebrates adapted to harness or consume this productivity. Higher still move fish and octopuses that prey upon smaller animals. Each transfer of energy incurs loss as heat, each step dependent on the stability of chemical supply from below. What overturned earlier assumptions was not merely the existence of these organisms, but the recognition that complex ecosystems could assemble without any input from sunlight. The idea that large animals required an indirect connection to photosynthetic life had shaped ecological thought. Vents revealed an alternative pathway, one in which Earth's internal heat substituted for stellar radiation. The biosphere was not confined to the thin, illuminated skin of the planet, but extended into darkness where rock and water interacted. Chemosynthetic pathways are ancient in evolutionary terms. Some scientists propose that early life may have relied on similar reactions long before oxygen accumulated in the atmosphere. The vent communities offer a living demonstration of that possibility, a modern expression of metabolism driven by redox chemistry rather than light capture. Enzymes within these bacteria channel electrons with precision, building carbon frameworks molecule by molecule. In this arrangement, energy does not descend from above. It rises, transformed through microbial labor into tissue and movement. The absence of sunlight does not imply absence of structure. Instead, a different architecture of life unfolds, anchored in chemical exchange. Where hydrogen sulfide meets oxygen in measured proportion, cells harvest power, and an ecosystem assembles itself around invisible currents of reaction. Not all danger announces itself with heat. Some of it drifts unseen through water, dissolved and silent, entering tissues without warning. Around hydrothermal vents, survival depends not only on tolerating temperature and pressure, but on enduring chemistry that would swiftly silence most surface life. Hydrogen sulfide is one such presence. In many animals, it interferes with cytochrome C oxidase, a key enzyme within mitochondria responsible for cellular respiration. When this pathway is blocked, oxygen cannot be used efficiently to produce energy. Cells starve, even when oxygen is abundant. For humans and other familiar creatures, even low concentrations can prove fatal. Yet vent organisms move within waters laced with this compound. Some avoid it through positioning, inhabiting narrow zones where concentrations are diluted. Others embrace it indirectly through symbiosis. In giant tube worms, hemoglobin binds hydrogen sulfide in a controlled manner, transporting it safely to internal bacteria. This binding prevents the molecule from interacting destructively with sensitive enzymes. The blood itself becomes a mediator, managing toxicity through reversible chemical associations. Beyond sulfide, vent fluids carry dissolved metals leached from basalt and other crustal rocks. Iron, copper, zinc, and traces of manganese circulate in elevated amounts. When superheated fluids mix with seawater, some metals precipitate into fine particles, yet significant quantities remain dissolved. Excess metals can generate reactive oxygen species within cells, damaging proteins, lipids, and DNA through oxidative stress. Vent animals counter this threat with specialized proteins known as metallothioneins. These small, cysteine-rich molecules bind heavy metals tightly, sequestering them in forms that reduce reactivity. By controlling intracellular metal concentrations, cells prevent toxic cascades. Antioxidant enzymes such as superoxide, dismutase, and catalase further neutralize reactive species, maintaining balance within a chemically active environment. Acidity presents another challenge. Vent effluent often has lower pH than surrounding seawater, reflecting dissolved carbon dioxide and other acidic components. Fluctuating pH can disrupt membrane potentials and enzyme function. Many vent species regulate internal acidity through ion transporters embedded in cellular membranes. These proteins actively exchange hydrogen ions for sodium or other cations, stabilizing cytoplasmic conditions despite external variation. Gill structures and other exchange surfaces display adaptations as well. Mucus layers can slow diffusion of harmful substances, creating microenvironments where concentrations are moderated before reaching epithelial cells. Some crustaceans possess thickened cuticles that limit direct exposure, while still allowing necessary gases to pass. The balance is delicate, preserving permeability for oxygen while restricting entry of damaging compounds. At the cellular level, heat shock proteins play a quiet but vital role. Although named for thermal stress, these chaperone molecules respond to chemical stress as well. They assist in refolding damaged proteins and targeting irreparable ones for degradation. In vent species, elevated baseline expression of such proteins provides constant readiness, a standing defense against unfolding threats. Together, these mechanisms form a layered system of protection. Blood chemistry neutralizes sulfide, binding proteins sequester metals, enzymes detoxify reactive byproducts, membranes regulate ion flow to preserve internal equilibrium. None of these strategies eliminates toxicity entirely. Instead, they transform lethal chemistry into manageable exposure. Life here does not ignore danger, it integrates it. Compounds that would end respiration in other animals become substrates, signals, or resources within adapted physiology. Cells adjust their internal landscapes to mirror the instability outside. In doing so, vent organisms reveal that survival at the edge of toxicity depends not on invulnerability, but on intricate control, a choreography of molecules maintaining coherence amid constant chemical flux. There is a boundary no larger than a handspan where existence steadies itself. On one side, fluid surges upward at temperatures that would dismantle living tissue in an instant. On the other, surrounding seawater presses in, cold and dense. Between these extremes lies a shifting corridor measured in centimeters, sometimes millimeters, where animals gather and hold their ground. Hydrothermal vents do not radiate uniform heat. Instead, they create intricate thermal layering. Superheated fluid emerges from narrow openings and begins to mix immediately with ambient water. The process is turbulent yet structured. Hot plumes rise, curl, and fragment. Cooler currents slip downward along chimney walls. Within this motion, steep gradients form, bands of temperature that change rapidly across short distances. A probe moved slowly through such a region records astonishing variation. At one point, it may register over 100 degrees Celsius, then a few centimeters away, only 10 or 20. These gradients are not static. They waver with fluctuations in flow, with subtle shifts in pressure and mineral deposition. The environment resembles a constantly redrawn map whose borders move by fractions of an inch. Animals do not anchor themselves within the hottest stream. Instead, they occupy surfaces where mixing has moderated extremes. A crab positions its body against rock that conducts some warmth, but is bathed intermittently in cooler currents. A worm extends its anterior into gentler water while keeping its posterior nearer chemical supply. Mussels cluster where convection delivers nutrients without overwhelming their tissues. Each organism selects a microhabitat defined by balance rather than maximum exposure. Thermal tolerance in multicellular life has limits. Proteins begin to denature as heat disrupts the hydrogen bonds and hydrophobic interactions that maintain their folded shapes. Membranes become too fluid, compromising integrity. Enzymatic reactions accelerate beyond optimal rates, leading to instability. Although vent species possess enhanced heat resistance, they remain constrained by biochemical boundaries. Survival depends not on enduring 400 degrees, but on remaining just outside that lethal threshold. Chimney structures themselves contribute to this moderation. Mineral walls conduct heat outward while also serving as barriers that slow direct contact with venting fluid. Porous textures create tiny eddies where hot and cold waters blend gradually. Within crevices and depressions, localized pockets of tolerable temperature persist. These niches can be narrower than a finger, yet stable enough to support dense aggregations. Behavior reinforces this fine-scale control. Many vent animals adjust their orientation continually. A slight shift of a claw, a subtle contraction of a body column, alters exposure. Some species display rapid withdrawal responses when encountering sudden bursts of heat, retreating deeper into protective tubes or behind shell valves. Others extend exploratory appendages to sample conditions before advancing. Such movements are small but decisive, guided by sensory systems attuned to thermal change. Even within a single organism, internal gradients mirror those outside. Circulatory systems distribute heat absorbed at one surface, preventing localized damage. Countercurrent exchange mechanisms in some species allow cooler blood returning from peripheral tissues to absorb warmth from outgoing blood, smoothing temperature differences before they reach vital organs. Physiology and environment interlock in a continuous exchange. Life at the edge of 400 degrees is therefore not a tale of defiance against fire, but of precision. The narrow band where chemistry is accessible and temperature is survivable shifts moment by moment. Within that band, creatures arrange themselves with remarkable accuracy, inhabiting a world defined not by vast expanses, but by the delicate space between too hot and just enough. Weight is rarely visible, yet here it defines everything. Above the vent fields lies not open sky, but kilometers of seawater, each layer adding its measure to the total load. At depths of 2,500 meters and beyond, pressure exceeds 250 atmospheres, more than 250 times what is felt at the surface. Every square centimeter of an organism's body bears this force continuously, without pause or relief. Pressure acts at the molecular scale, long before it alters visible form. Proteins, those intricately folded chains of amino acids, depend on precise spatial arrangements to function. Their three-dimensional shapes arise from delicate balances between hydrogen bonds, hydrophobic interactions, and electrostatic forces. Elevated pressure can shift these balances. Cavities within proteins compress. Water molecules are driven into spaces they would not normally occupy. Conformations change subtly, sometimes enough to reduce activity or destabilize the structure entirely. Enzymes from surface species often lose efficiency under such compression. Reaction rates alter as active sites deform. Some proteins unfold partially, exposing hydrophobic regions that trigger aggregation. In shallow water organisms, these changes accumulate rapidly at depth. Cellular machinery falters, not because of heat or toxins, but because physical force distorts the architecture of life itself. Membranes face a related challenge. The lipid bilayer that encloses each cell must remain fluid enough to allow embedded proteins to function, yet cohesive enough to maintain integrity. High pressure tends to order lipid molecules more tightly, decreasing membrane fluidity. If membranes become too rigid, transport proteins cannot change shape effectively, ion gradients dissipate, and communication between cell and environment degrades. Vent organisms counter this compression through biochemical adjustments. Their membranes often contain higher proportions of unsaturated fatty acids, whose kinks prevent tight packing even under pressure. Some species incorporate unique lipid structures that maintain optimal fluidity across wide pressure ranges. Proteins may exhibit reduced internal cavities and enhanced surface interactions, features that resist deformation. Small organic molecules, known as piezolites, accumulate within cells, stabilizing protein structure by offsetting pressure-induced changes in hydration. For animals accustomed to surface conditions, descent into such depths would be catastrophic. Gas-filled spaces within bodies would compress drastically, collapsing lungs or swim bladders. Even without air spaces, cellular processes would slow or fail as enzymes encountered unfamiliar compression. Neural transmission, muscle contraction, and metabolic pathways would falter almost immediately. Survival requires not temporary tolerance but lifelong adaptation encoded into every membrane and molecule. At hydrothermal vents, pressure does not act alone. It intersects with heat and chemistry, creating combined stresses that amplify one another. Elevated temperature tends to increase membrane fluidity, while pressure decreases it. In some respects, these forces counterbalance, yet their interaction is complex. Proteins stabilized against heat may respond differently under simultaneous compression. The biochemical toolkit of vent species reflects this dual challenge, shaped by selection in an environment where extremes coexist. Development from larval stages onward occurs under these conditions, ensuring that structural features are integrated rather than imposed. Cellular components assemble in a high-pressure context, folding and organizing in ways compatible with compression. What would seem extraordinary from the surface becomes ordinary here, a background constant against which life unfolds. Pressure remains unseen, without color or movement, yet it permeates every tissue. It presses inward from all directions, reshaping molecules, influencing reactions, guiding evolutionary pathways. In the quiet darkness of the deep, this hidden extreme stands beside heat and chemistry, an ever-present force that life has learned not to resist, but to incorporate into its very design. The seafloor here is not a fixed stage, but a shifting manuscript written in mineral and erased by fire. What seems established, crowded with living forms, rests upon crust that is still being born. Hydrothermal vents are transient expressions of deeper forces, their existence tied to fractures that may open and close within a human lifetime. Along mid-ocean ridges, tectonic plates separate by mere centimeters each year, yet that slow divergence reshapes entire landscapes. Magma ascends to fill the gap, cooling into new basalt. Seawater infiltrates cracks, descends, heats, and rises again, constructing chimneys from precipitated minerals. As long as circulation continues, towers grow and chemical gradients persist. When pathways shift or seal, the flow weakens. Heat fades. Surfaces that once shimmered with activity cool into inert stone. Volcanic eruptions can alter this terrain in hours. Lava may spill outward in thick sheets, burying established colonies beneath fresh rock. Fluid routes are rerouted, through newly fractured crust, abandoning older outlets. Chimneys that once supported dense assemblages are entombed or severed from their energy source. Organisms attached to their surfaces have little warning. The event is geological, not biological, yet its consequences are immediate for every creature bound to that site. Even without eruption, chimneys are inherently unstable. They form from brittle mineral deposits, often riddled with internal channels. As deposits accumulate unevenly, structural weaknesses develop. A slight change in flow, a subtle tremor in the crust, can cause collapse. Sections shear away and tumble downslope. Animals that relied on specific gradients are displaced into unsuitable water, where temperature or chemistry no longer align with their needs. Entire communities can vanish within days. The tube dwellers that anchored themselves in careful positions, the crustaceans that navigated narrow bands, the microbial films that formed the foundation of the system, all may be extinguished at a single location. The deep sea does not cushion such loss with gradual decline. Change can be abrupt and comprehensive. Yet this impermanence has shaped life histories in profound ways. Many vent species mature rapidly, reaching reproductive capability in shorter spans than relatives in more stable habitats. Energy derived from chemical flux allows accelerated growth, enabling organisms to reproduce before habitat shifts. High fecundity increases the probability that offspring will encounter newly active sites elsewhere along the ridge. Larvae released into the water column drift with deep currents, sometimes for weeks. During this pelagic phase, they remain suspended between destruction and opportunity. Chemical cues dispersed through seawater guide settlement, subtle traces of reduced compounds, or slight thermal anomalies signal proximity to an emerging vent. When such cues align with intolerable limits, larvae descend and attach, beginning the process of colonization anew. Genetic analyses reveal connectivity across vast distances, indicating that populations are linked through episodic dispersal. A vent field extinguished in one region does not necessarily mean the end of its inhabitants as a species. Persistence arises from movement across a mosaic of habitats, some waning while others ignite. Succession unfolds in recognizable stages. Microorganisms colonize bare mineral surfaces first, establishing biofilms that modify local chemistry. Small grazers arrive, followed by larger invertebrates and predators. Complexity builds over years, sometimes decades, until another geological pulse resets the sequence. Destruction and renewal are interwoven, neither final nor absolute. In this environment, survival is not measured by permanence, but by continuity through change. Life endures not by clinging to a single chimney, but by dispersing, reproducing, and adapting to instability. The vent field may vanish overnight, yet the broader system persists, sustained by the restless crust and the quiet resilience of organisms shaped to expect impermanence. Between one vent field and the next lies a vast expanse of dark seafloor, cold and seemingly empty. No glowing plumes mark the way. No continuous chain of habitat connects one oasis to another. The distances can stretch for hundreds of kilometers along a mid-ocean ridge, interrupted only by bare basalt and sediment. Yet life moves across these intervals, carried not by intention, but by currents. Most vent animals begin as larvae, released into the surrounding water in astonishing numbers. These early stages are small, often translucent, equipped with cilia or tiny appendages that allow limited movement. They do not crawl along the bottom as their parents do. Instead, They rise into the water column and enter the slow circulation of the deep ocean. There, they drift for days, sometimes weeks, suspended within layers of moving water that trace invisible pathways along the ridge. Currents at depth are subtle but persistent. They follow contours of topography, guided by differences in temperature and salinity. Larvae become passengers within these flows, dispersed outward from their birthplace. Many will never encounter another active site. Some will settle in unsuitable terrain and perish. Yet a fraction continue onward, borne by circulation patterns that link vent fields separated by great distance. During this journey, larvae are not entirely passive. Many possess sensory structures capable of detecting chemical cues. Dissolved compounds associated with hydrothermal activity, traces of reduced sulfur, or faint thermal anomalies create gradients in the water. As larvae approach such signals, their swimming behavior can change. They may descend toward the seafloor, exploring with brief bursts of motion. Settlement occurs only when conditions fall within precise limits of temperature and chemistry. The ability to sense these cues is critical because vents are patchy and ephemeral. A larva settling at the wrong site may find no energy source sufficient to sustain growth. Selection, therefore, favors individuals responsive to specific combinations of dissolved molecules and thermal signatures. Laboratory experiments have shown that certain vent species increase settlement rates when exposed to water conditioned by active vents, indicating a refined sensitivity to environmental chemistry. Despite the immense separation between fields, genetic studies reveal continuity among populations. Analysis of mitochondrial DNA and other genetic markers shows that individuals from distant vents often share recent common ancestry. This pattern suggests ongoing exchange through larval dispersal. Ocean currents, though slow, provide enough connectivity to prevent complete isolation. Over evolutionary time, this movement maintains genetic diversity and resilience. Not all species disperse equally. Some have larvae that remain in the plankton longer, extending their potential range. Others settle quickly, leading to more localized populations. The balance between dispersal and retention shapes community structure along each ridge segment. In regions where vents are closely spaced, exchange may be frequent. Where gaps widen, only the most enduring larvae bridge the distance. This system of drift and detection allows life to track geological change. When a new vent opens, its chemical plume begins to mix with surrounding water. Larvae passing within range encounter altered gradients and respond. Gradually, pioneers arrive, attach, and begin to grow. Over time, a community assembles upon what was once bare rock. Across the dark stretches between boiling waters, continuity is maintained not by permanence of place, but by movement through space. Larvae drifting in deep currents carry the future of their species, linking scattered habitats into a network sustained by chemistry and flow. Color arrives here not from sunlight, but from chemistry. Against the dim outline of the seafloor, some chimneys exhale plumes as dark as ink, while others release clouds the color of pale milk. These contrasts do not arise from light scattering alone, but from the minerals carried within each rising column. Black smokers earn their name from the dense, soot-like appearance of their emissions. The fluid issuing from these vents is typically hotter, often exceeding 350 degrees Celsius before mixing with surrounding seawater. As the superheated solution meets colder water, dissolved metals such as iron, copper, and zinc precipitate rapidly as sulfide minerals. Tiny particles form in suspension, creating a dark plume that billows upward before dispersing. The chimney walls themselves become layered with these metal sulfides, building tall, brittle structures composed largely of pyrite, chalcopyrite, and sphalerite. White smokers, by contrast, emit fluids that are generally cooler, often in the range of 200 to 300 degrees Celsius before dilution. Their pale appearance results from different mineral chemistry. Instead of iron-dominated sulfides, these vents release higher concentrations of barium, calcium, and silicon. When these elements combine with sulfate in seawater, they form light-colored minerals such as barite and anhydrite. The resulting plume appears gray to white, softer in tone, though no less dynamic in motion. Temperature differences influence not only mineral composition, but also the surrounding biological community. Black smokers, with their higher thermal output and greater concentrations of certain metals, tend to support species adapted to steeper chemical gradients. Organisms that rely on abundant reduced compounds often cluster near these structures, where energy flux is intense. Some shrimp species form dense aggregations along the flanks of black smoker chimneys, positioning themselves where chemical supply remains high yet tolerable. White smokers often host communities with slightly different arrangements. Because their fluids are somewhat cooler and mineral composition distinct, the gradients they create can favor alternative assemblages of mussels, worms, and crustaceans. Microbial populations also vary, reflecting differences in available substrates for metabolism. Even subtle shifts in temperature and chemistry alter which bacterial strains dominate surfaces and internal symbioses. Visually, the two types of vents offer contrasting scenes. Black smokers can rise as dark towers, their plumes thick and opaque, creating the impression of smoke pouring into water. The surrounding mineral deposits may appear metallic, with shimmering surfaces reflecting faint light from submersibles. White smokers appear more chalky, their chimneys often lighter in color, sometimes more fragile in texture. Their plumes spread like cloudy veils rather than dense columns. Despite these differences, both forms arise from the same underlying process of seawater circulating through heated crust and returning enriched with dissolved material. Variations in rock composition, temperature of underlying magma, and depth of circulation shape the final chemistry of the venting fluid. Small changes in subsurface pathways can transform one style of vent into another over time. Communities near each type are not rigidly separated. Many species tolerate a range of conditions and may occupy zones where black and white smokers occur in proximity. Yet the subtle distinctions in mineral output and thermal structure create microhabitats that influence distribution patterns across a vent field. In this landscape of darkness, color is born from elements rather than light. Black and white smokers stand as parallel expressions of the planet's internal chemistry, their plumes carrying different signatures into the sea. Around them, life arranges itself in response to those signatures, adapting to nuances of temperature and mineral flow that define each chimney's character. Far below the drifting continents, the planet continues a quiet act of renewal. A seam runs along the ocean basins, stretching for tens of thousands of kilometers, marking where tectonic plates draw apart. These mid-ocean ridges are not mountains in the familiar sense, yet they form the longest continuous volcanic chain on Earth. Along their crest, new crust emerges as molten rock ascends from the mantle and cools into basalt. Seafloor spreading unfolds at a pace measured in centimeters each year. The movement is slow, almost imperceptible, yet over millions of years it reshapes entire oceans. As plates separate, pressure on underlying mantle decreases, allowing partial melting to occur. Magma rises through fractures, filling the gap, solidifying as it meets colder surroundings. This constant creation of crust sustains the architecture of the ridge system. Within this restless zone, seawater finds entry. Fractures and porous basalt permit cold ocean water to descend into the crust. As it travels downward, it approaches regions heated by newly formed magma chambers. Temperatures increase steadily. The water interacts chemically with surrounding rock, dissolving metals and altering mineral structures. Heated and transformed, it becomes buoyant and begins its ascent back toward the seafloor. The circulation resembles a vast convective engine. Cold water sinks, absorbs heat, and returns upward as a plume of altered fluid. Where it re-enters the ocean, hydrothermal vents form. The energy driving this exchange originates not from the sun, but from the residual heat of planetary formation and the ongoing decay of radioactive elements within the mantle and crust. Uranium, thorium, and potassium isotopes release energy as they decay, contributing to Earth's internal heat budget. Although the total heat flux from Earth's interior is modest compared to solar input at the surface, it is steady and global. Estimates suggest that the planet releases tens of terawatts of heat through conduction, volcanic activity, and hydrothermal circulation. Mid-ocean ridges account for a significant fraction of this output. The hydrothermal systems associated with them transfer heat from magma to seawater efficiently, cooling newly formed crust while sustaining chemical gradients in the deep ocean. This internal furnace does more than build rock. It drives geochemical cycles that influence ocean composition over geological time. As seawater circulates through hot basalt, magnesium is removed and metals are added. Altered fluids returning to the ocean contribute to the balance of elements that marine life depends upon. In this way, the ridge system participates in regulating the chemistry of the global ocean. Beneath each active vent field lies a network of cracks and magma bodies, their arrangement shifting as spreading continues. Some regions experience more vigorous volcanic episodes, others quieter phases dominated by steady circulation. The pattern is neither uniform nor static. It reflects the dynamics of mantle convection and plate motion, processes operating on scales both vast and intimate. The existence of life around vents is therefore inseparable from plate tectonics. Without seafloor spreading, magma would not approach the surface in this manner. Without magma, there would be no heating of descending seawater, no emergence of mineral-rich fluids. The ecosystems described earlier trace their origins to this deeper architecture, rooted in the slow divergence of plates and the heat retained within the planet. In the darkness of the abyss, the ridge crest glows not with visible flame, but with thermal exchange. Earth's interior, still warm from its formation and sustained by radioactive decay, continues to release energy. That energy moves through rock into water and outward into the ocean, sustaining systems that rely on chemistry rather than light. The deep sea's internal furnace burns without spectacle, yet its influence shapes crust, ocean, and life alike. In the late 20th century, long before vent fields were mapped in detail, the deep ocean was still imagined as a broad and quiet plain. Oceanographers understood plate tectonics was reshaping the seafloor, yet the abyss itself remained largely unseen. The technology required to reach such depths was rare, delicate, and experimental. Each descent carried uncertainty, not only about what might be found, but about whether the vehicle would return intact. In 1977, the submersible Alvin prepared for a dive near the Galapagos Rift. Built to withstand crushing pressure, Its titanium sphere carried a small crew into darkness few humans had entered. Through narrow viewports, observers watched sediment drift past as the craft descended thousands of meters. Their mission centered on geology, on measuring heat flow and examining volcanic formations associated with spreading ridges. As Alvin approached the seafloor, its lights revealed basaltic terrain fractured by tectonic strain. Instruments recorded elevated temperatures in certain areas, confirming the presence of hydrothermal activity predicted by theory. What theory did not predict was what lay beyond the rock itself. In the glow of artificial light, pale structures appeared, rising from the crust in clustered formations. Closer inspection showed these were not purely mineral constructs. Around the vent openings stood dense communities of organisms, many unfamiliar to science. Long white tubes crowned with red plumes, swarms of shrimp, clumps of mussels and crabs, all gathered where superheated fluid met cold seawater. The crew had expected barren geology. Instead, they encountered an ecosystem thriving without sunlight. Initial reactions combined excitement with disbelief. Prevailing biological models held that complex life depended ultimately on photosynthesis. Sunlight fueled primary production, which in turn supported higher trophic levels. The deep sea, lacking light, was thought to rely on a slow rain of organic debris sinking from above. The abundance observed at the vent contradicted that assumption. Here was biomass concentrated around a source of chemical energy rather than solar input. Samples collected by Alvin and subsequent expeditions confirmed that these organisms were not transient visitors. They were specialized species adapted to this environment. The discovery forced a reassessment of ecological principles. Chemosynthesis, previously known in limited microbial contexts, was recognized as the foundation of a robust food web. Energy could enter biological systems through pathways entirely independent of the sunday. The historical context magnified the impact. Plate tectonics itself had only recently gained broad acceptance within geology. The idea that spreading ridges might host dynamic hydrothermal systems was still being tested. The sight of living communities intertwined with active volcanism unified geological and biological processes in a vivid way. Earth's internal heat was not merely shaping crust, it was sustaining ecosystems. Public imagination was also stirred. Images and descriptions from the dives circulated through scientific journals and media reports. The notion of life flourishing in darkness, sustained by chemicals leaching from rock, expanded the perceived boundaries of habitability. Researchers began to consider whether similar systems might exist elsewhere in the solar system, beneath icy crusts or within subsurface oceans. Within the scientific community, debate and revision followed. Textbooks were updated. Models of deep-sea ecology incorporated vent systems as distinct biomes. Funding increased for further exploration, leading to the discovery of additional fields along other ridges and back-arc basins. Each expedition added detail, yet the initial moment of recognition retained its clarity. The first scientists who saw these communities did more than document an anomaly. They witnessed a shift in understanding, a redefinition of where and how life could persist. Through a small viewport in a steel sphere, beneath kilometers of water, established assumptions gave way to a broader view of Earth's possibilities. Long before animals anchored themselves to chimneys, before even the first cell divided, the young Earth was a different world. Continents were smaller, oceans warmer, the atmosphere lacking oxygen. Volcanic activity was more widespread, and the crust thinner, allowing heat from the mantle to interact readily with seawater. In that ancient setting, hydrothermal systems may have been far more common than they are today. The origin of life remains one of science's most enduring mysteries, yet hydrothermal vents have become central to one influential hypothesis. Rather than life beginning in a sunlit tidal pool, some researchers propose it emerged in the chemically rich interfaces where hot, mineral-laden fluids met cooler ocean water. In such environments, steep gradients in temperature, pH, and chemical composition would have existed across microscopic distances. Life as we know it relies on gradients. Cells maintain differences in proton concentration across membranes, using that imbalance to drive the synthesis of adenosine triphosphate, the molecule that powers metabolism. Hydrothermal vents naturally generate proton gradients as alkaline fluids mix with more acidic seawater. Within porous mineral structures, tiny compartments form where these differences can persist, resembling primitive versions of cellular boundaries. Mineral surfaces themselves may have acted as catalysts. Iron-sulfur minerals, common in vent systems, can facilitate redox reactions similar to those used by modern enzymes. In laboratory experiments, such minerals promote the formation of simple organic molecules from inorganic precursors under conditions that approximate early Earth. These reactions suggest that geochemistry could have provided a scaffold for increasingly complex chemistry. Early oceans likely contained higher concentrations of dissolved metals and reduced compounds than today. Without abundant oxygen to react with them, molecules such as hydrogen, methane, and ammonia may have accumulated. In the presence of heat and catalytic minerals, these substances could assemble into amino acids, nucleotides, and other building blocks of life. Over time, networks of reactions might have become self-sustaining, giving rise to the first metabolic pathways. The vent hypothesis also addresses the challenge of stability. Surface environments on the young Earth were subject to intense ultraviolet radiation, meteor impacts, and fluctuating temperatures. In contrast, hydrothermal systems provided continuous energy and relative protection beneath the ocean surface. Within mineral pores, organic molecules could concentrate rather than dispersing into vast waters. Concentration increases the likelihood of interaction, allowing complex structures to emerge. Some models envision early life not as free-floating cells, but as communities embedded within mineral matrices. Over evolutionary time, lipid membranes may have formed, enclosing metabolic reactions and enabling independence from the rock substrate. The universal presence of iron-sulfur clusters in modern enzymes has been cited as a molecular echo of these origins, hinting at ancient ties between biology and vent geochemistry. Not all scientists agree with this scenario. Competing hypotheses propose shallow water settings, or even extraterrestrial delivery of organic compounds. Yet the vent model remains compelling because it unites energy source, catalytic surfaces, and chemical gradients within a single environment. It frames life as an outgrowth of planetary processes rather than an isolated anomaly. When considering organisms that now thrive around modern vents, one can glimpse a distant parallel. They rely on chemical disequilibrium, on reactions between reduced and oxidized compounds. Their metabolism echoes what early biochemical systems might have exploited. Though separated by billions of years, the connection between rock, water, and energy persists. Whether life began precisely in such settings cannot yet be proven. Still, hydrothermal systems offer a coherent narrative in which Earth's internal heat and ocean chemistry converge to foster complexity. In the quiet interplay of minerals and gradients, the first stirrings of metabolism may have found a foothold, anchoring biology to the restless heart of the planet. Before animals ever anchored themselves to mineral walls, life here existed as something far smaller and more ancient. Within the superheated circulation of water through rock, microscopic organisms established metabolisms tuned to extremes. These microbes form the unseen foundation of vent systems, inhabiting crevices, fluids, and mineral grains where conditions approach the upper boundaries of biology. Among them are members of the domain Archaea, a lineage distinct from bacteria and eukaryotes. Though superficially similar to bacteria in size and shape, Archaea possess unique membrane lipids and genetic machinery that set them apart. Many vent-associated archaea are hyperthermophiles, organisms whose optimal growth temperatures exceed 80 degrees Celsius. Some continue dividing at temperatures near or above 100 degrees under high pressure, where water remains liquid. Their enzymes reveal how such survival is possible. Proteins within hyperthermophilic archaea display structural features that confer remarkable thermal stability. Compared to enzymes from temperate organisms, they often contain increased numbers of ionic bonds and tighter packing within their cores. These characteristics reduce flexibility at lower temperatures but prevent unfolding at higher ones. Hydrogen bonds and salt bridges reinforce the folded state, resisting the disruptive motion that heat induces. Membrane composition also differs. Instead of ester-linked lipids common in bacteria and eukaryotes, many archaea possess ether-linked lipids with branched isoprenoid chains. Some form monolayer membranes rather than bilayers, creating a more rigid barrier less prone to heat-induced leakage. These chemical distinctions allow cells to maintain internal order despite external temperatures that would compromise typical membranes. Laboratory studies have isolated strains from vent fluids and cultivated them under controlled conditions that mimic high pressure and temperature. In pressurized bioreactors, researchers observe growth at temperatures exceeding 110 degrees Celsius for certain species. Such findings extend the known thermal limits of life and challenge assumptions about protein stability. At sea level pressure, water would boil at these temperatures, yet under compression, liquid conditions persist, enabling metabolism to proceed. Genomic analyses of hyperthermophiles reveal genes encoding chaperone proteins that assist in folding newly synthesized enzymes and repairing those partially denatured. DNA-binding proteins stabilize genetic material, protecting it from heat-induced strand separation. Specialized reverse gyrase enzymes introduce positive supercoils into DNA, increasing its resistance to thermal unwinding. These molecular adaptations illustrate how deeply integrated heat tolerance is within cellular systems. The upper thermal boundary for life remains an active area of research. Experiments suggest that beyond certain temperatures, the stability of fundamental biomolecules becomes untenable. Even with pressure-enhancing liquid water stability, chemical reactions that degrade amino acids and nucleotides accelerate at extreme heat. Current evidence places the highest confirmed growth temperatures for organisms slightly above 120 degrees Celsius under laboratory conditions, though natural environments sustaining such levels are rare. Vent ecosystems host gradients where hyperthermophiles occupy the hottest niches, often within porous rock or in mixing zones close to fluid exit points. As temperature decreases outward, mesophilic and thermophilic bacteria and archaea replace them, forming layered microbial communities structured by thermal preference. This arrangement mirrors the broader pattern seen in larger organisms, yet at a scale measured in micrometers. The study of extremophile microbes has influenced biotechnology as well. Heat-stable enzymes isolated from thermophiles have proven valuable in industrial processes requiring elevated temperatures. Their resilience under harsh conditions reduces contamination risk and increases efficiency. Thus, adaptations forged in deep ocean vents find application far beyond their original habitat. In these microorganisms, life approaches its thermal frontier. Their existence demonstrates that the limits of biology extend deeper into heat than once imagined. Within the mineral pores of the seafloor, archaea and bacteria persist where rock and water converge, sustaining metabolism in conditions that once seemed incompatible with living systems. Heat defines a boundary that multicellular life approaches carefully. Unlike microbes that inhabit microscopic pores and divide rapidly, animals and plants consist of coordinated tissues, each dependent on precise molecular choreography. As temperature rises, that choreography becomes increasingly difficult to maintain. The stability of proteins, membranes, and genetic material sets an upper threshold beyond which complexity begins to unravel. Proteins are central to this limit. Every muscle contraction, nerve impulse, and metabolic reaction relies on enzymes folded into specific three-dimensional forms. These shapes are stabilized by hydrogen bonds, ionic interactions, and hydrophobic forces that hold amino acid chains in place. Elevated temperature increases molecular motion, shaking these interactions. When enough bonds break, a protein unfolds, losing its functional shape. This process, known as denaturation, can be reversible at modest temperatures, yet becomes permanent once structural damage accumulates. In unicellular organisms, damaged proteins can sometimes be degraded and replaced quickly, allowing survival near extreme heat. Multicellular animals face greater constraints. Specialized tissues such as nerves and reproductive organs are particularly sensitive to temperature fluctuations. Coordination among cells requires consistent membrane potentials and signaling pathways, both of which depend on intact protein structures. Sustained exposure to high temperatures disrupts these systems, leading to systemic failure. Laboratory experiments illustrate the distinction between brief and prolonged exposure. Many marine invertebrates can tolerate short bursts of temperatures well above their normal range. Heat shock proteins activate rapidly, binding to partially unfolded enzymes and assisting in refolding. During transient spikes, this protective response may preserve function. However, when exposure extends beyond minutes into hours, repair mechanisms are overwhelmed. Accumulated denaturation and membrane destabilization impair essential processes. For most multicellular animals, sustained survival above 50 degrees Celsius is rare. Some vent-associated worms and crustaceans endure transient contact with water approaching or exceeding that level, yet internal body temperatures remain moderated by positioning within gradients. Recorded maximum tolerances for brief exposure in certain species reach near 60 degrees Celsius, though these instances often involve seconds rather than prolonged habitation. Experimental studies on thermophilic invertebrates have measured lethal temperature thresholds under controlled conditions. When held at constant elevated temperatures, survival times decrease sharply as limits are approached. A difference of a few degrees can determine whether an organism survives for hours or succumbs within minutes. The relationship between temperature and time forms a curve rather than a single point, emphasizing that tolerance depends on duration as well as intensity. Cell membranes impose additional constraints. As temperature rises, lipid bilayers become increasingly fluid, compromising barrier function. Ion gradients dissipate and embedded proteins lose optimal orientation. While some vent species adjust membrane composition to enhance stability, there remains a point beyond which fluidity disrupts cellular integrity. DNA itself, though relatively stable compared to proteins, can suffer damage through accelerated chemical reactions at high temperatures. Pressure in deep environments modifies these limits slightly by stabilizing certain molecular interactions, yet it does not eliminate them. Multicellular life appears bounded by the cumulative fragility of its coordinated systems. The complexity that allows specialization and movement also introduces vulnerability when thermal energy becomes excessive. The highest confirmed sustained growth temperatures for multicellular organisms remain well below those of hyperthermophilic microbes. Life at vent fields demonstrates that animals can approach these boundaries through behavioral positioning and biochemical adaptation, yet they inhabit margins rather than extremes. Their survival records reflect careful negotiation with heat, not immunity to it. Thus, the temperature limit of multicellular life is not defined by a single dramatic number, but by the interplay between molecular stability and exposure time. Within that interplay, organisms find narrow windows where structure holds, reactions proceed, and coordinated life persists at the edge of thermal possibility. Not all chemical oases in the deep are born of scalding water. In some regions of the seafloor, the chemistry rises without dramatic heat, seeping upward through sediments in a slow and steady breath. These places are known as cold seeps, though the term refers only to their temperature relative to hydrothermal vents. The fluids emerging here are often near ambient deep sea conditions, yet they carry compounds capable of sustaining life in similar ways. Cold seeps commonly release methane and hydrogen sulfide from beneath the seabed. Methane may originate from the breakdown of organic matter buried over millions of years, or from deeper thermogenic processes within the crust. As this gas migrates upward, it dissolves into surrounding water or forms bubbles that percolate through sediment. Unlike the turbulent plumes of hot vents, seep emissions are subtle, sometimes visible only as shimmering distortions or clusters of gas escaping from the seafloor. Within these sediments, bacteria and archaea perform chemosynthesis much as they do near vents, though the energy source differs. Methanotrophic microbes oxidize methane, converting it into carbon dioxide while generating usable energy. In other cases, consortia of archaea and sulfate-reducing bacteria collaborate in a process known as anaerobic oxidation of methane. Together, they transform methane and sulfate into bicarbonate and sulfide, establishing a chemical foundation for higher organisms. The result is a community structure reminiscent of vent systems, yet shaped by gentler gradients. Tube worms, mussels, and clams gather at seep sites, many hosting symbiotic bacteria within their tissues. These symbionts metabolize methane or sulfide supplied by the seep, providing nourishment to their hosts. Although the absence of extreme heat alters the physical environment, the underlying principle remains the same. Chemical disequilibrium drives primary production in darkness. Temperature differences influence both physiology and community composition. At cold seeps, organisms are not challenged by rapid thermal fluctuations or proximity to superheated fluid. Protein stability constraints are therefore less severe, and species need not endure brief encounters with near-lethal temperatures. Instead, adaptation focuses on tolerating elevated concentrations of methane and sulfide, along with the often soft, unstable sediments in which seeps occur. Carbonate structures frequently develop at seep sites, as bicarbonate produced by microbial metabolism reacts with calcium in seawater. These authigenic carbonates can form hard grounds that provide attachment surfaces for sessile organisms. Over time, seep fields may accumulate extensive carbonate mounds, creating habitats distinct from the mineral chimneys of hydrothermal systems. The pace of change at cold seeps is typically slower. While vents may flicker on and off with tectonic shifts, seeps can persist for centuries or longer if methane supply remains steady. This relative stability allows communities to mature gradually, supporting longer-lived species and more continuous assemblages. Nevertheless, they remain dependent on subsurface processes and can wane if fluid flow diminishes. From a biochemical perspective, cold seeps expand the concept of chemosynthetic ecosystems beyond high-temperature settings. They demonstrate that intense heat is not a prerequisite for life independent of sunlight. Instead, what matters is the presence of reduced compounds and oxidants in contact, creating gradients that microbes can exploit. In the quiet sediments where methane rises in slow pulses, life arranges itself around invisible currents of chemistry. The story mirrors that of hydrothermal vents, yet unfolds without towering plumes or mineral spires. Here, energy seeps rather than surges, and communities grow in the absence of both sunlight and fire, sustained by reactions occurring beneath the surface of the seafloor. Movement here is not limited to grazers and symbionts. Beyond the clustered tubes and shells, shapes glide through dim water, patient and deliberate. Predators inhabit these chemical oases as surely as producers and filter feeders do, weaving a network of pursuit and consumption that completes the ecology of the vent field. Among the most striking are vent octopuses, pale-bodied and supple, their skin often lacking the bold pigmentation seen in shallow relatives. They drift just above the seafloor, arms extended with quiet precision. Suckers probe rock and mineral surfaces, tasting chemical traces left behind by potential prey. Unlike fast-moving pelagic hunters, these octopuses move with measured intent, conserving energy in an environment where food is concentrated but localized. Their diet includes crustaceans, worms, and occasionally small fish that stray too close. With flexible arms and acute tactile sensitivity, they can extract animals from crevices or grasp prey that attempts escape. The absence of light has not diminished their capacity for complex behavior. Instead, their nervous systems rely on touch and chemosensory input, interpreting currents and subtle disturbances in water. Zooarcid fish, often called eelpouts, form another tier of predation. Elongated bodies allow them to navigate tight spaces among mineral formations. Their heads are broad, jaws equipped with small but effective teeth. They hover near clusters of invertebrates, ready to strike at shrimp or juvenile worms. Some species display reduced eyes, consistent with a habitat where vision offers limited advantage, yet their lateral line systems remain finely tuned to vibration. These predators occupy positions within a feeding network anchored in chemosynthesis. At the base lie microbes converting reduced compounds into biomass. Above them are primary consumers, grazing on bacterial films or hosting symbionts. Secondary consumers, such as shrimp and small crabs, feed on these grazers. Predators then feed on them, transferring energy upward through the system. The network is not strictly linear. Scavenging plays a significant role. When organisms die, their bodies become resources for opportunistic feeders. Crabs may tear apart remains, while smaller invertebrates consume fragments. Microbes decompose what is left, returning nutrients to the chemical cycle. In this way, matter circulates repeatedly within the community, minimizing waste in a setting where external input is limited. Vent octopuses contribute to this recycling indirectly. By capturing prey, They influence population sizes of smaller organisms, preventing any single species from overwhelming available resources. Zooarcid fish exert similar control. Predation introduces balance, distributing pressure across the community and maintaining diversity. Reproductive strategies among these predators reflect environmental constraints. Some vent octopuses lay eggs attached to stable substrates near active areas, ensuring that developing embryos remain within reach of chemical energy once hatched. Parental investment may be extended, with females guarding clutches for prolonged periods. Such behavior suggests adaptation to a habitat where suitable sites are patchy and valuable. The presence of predators underscores the maturity of vent ecosystems. These are not simple assemblages of microbes and filter feeders, but structured communities with multiple trophic levels. Energy captured from chemical reactions moves through bodies of increasing complexity, culminating in animals capable of sophisticated hunting strategies. In the stillness between pulses of venting fluid, a zooarcid waits, poised among rocks, while an octopus glides overhead, arms unfurling like slow currents. Their existence depends on the invisible work of microbes and the steady transfer of energy through countless interactions. Even here, in water shaped by heat from below, the ancient patterns of predation and balance persist, completing the circle of life in a realm without sunlight. Depth adds another layer of silence. As the seafloor drops farther from the surface, pressure increases and temperature of surrounding water hovers near freezing. Yet even in these remote basins, hydrothermal systems persist, marking the continuation of planetary heat release far below familiar thresholds. Some vent fields lie deeper than 5,000 meters, where darkness and compression define the setting with unyielding constancy. In the western Pacific Ocean, the Mariana Back Arc hosts some of the deepest confirmed hydrothermal vents. The site, known as the Champagne Vent, discovered in the early 21st century, lies at depths exceeding 3,500 meters, notable not only for its depth, but for fluids enriched in carbon dioxide that form shimmering droplets under pressure. Even deeper systems have been identified along segments of the Mariana Trench region, where venting occurs at nearly 5,000 meters, extending the known vertical reach of these ecosystems. Across the Atlantic Ocean, vent fields line the mid-Atlantic ridge, including sites such as Tag and Snake Pit. While many of these occur between 2,000 and 3,500 meters, exploration has revealed variation along the ridge axis, with some deeper basins hosting active circulation. The Atlantic discoveries were among the earliest after the initial Galapagos findings, confirming that hydrothermal activity was not confined to a single ocean, but distributed along global spreading centers. The Indian Ocean, long less explored, has also yielded significant vent fields. Along the central Indian Ridge and the southwest Indian Ridge, researchers have documented systems at depths approaching and exceeding 4,000 meters. These regions are characterized by slower spreading rates compared to parts of the Pacific, influencing the chemistry and structure of venting fluids. Distinct faunal assemblages occur here, including species not found in Pacific or Atlantic sites, suggesting evolutionary divergence shaped by geographic isolation. Polar regions add further complexity. In the Arctic Ocean, hydrothermal vents were discovered along the Gokul Ridge, one of the slowest spreading ridges on Earth. Some of these Arctic vents lie beneath permanent sea ice at depths greater than 4,000 meters. The overlying ice cover does not diminish vent activity, though it complicates exploration. Remotely operated vehicles deployed through ice-covered waters have revealed active chimneys and associated communities, demonstrating that tectonic heat release continues even in the coldest ocean basins. Antarctic discoveries have expanded the map southward. In the Southern Ocean, vents identified along the East Scotia Ridge occur at depths around 2,400 to 2,600 meters. These systems host unique species, including yeti crabs with setae that harbor symbiotic bacteria. Isolation by circumpolar currents appears to have limited species exchange with other oceans, resulting in distinctive assemblages adapted to local conditions. Depth records matter because they illustrate the adaptability of vent communities to extreme pressure combined with chemical and thermal gradients. At 5,000 meters, pressure exceeds 500 atmospheres, imposing additional constraints on cellular structures and physiological processes. The persistence of active ecosystems at such depths underscores the resilience of chemosynthetic life across a wide vertical range. Exploration of these deepest fields relies on advanced submersibles and remotely operated vehicles capable of withstanding immense compression. Each descent extends understanding of how vent systems vary with spreading rate, magma supply, and tectonic context. Differences in depth influence fluid chemistry, mineral deposition, and community composition, yet the underlying principle remains consistent. From tropical back-arc basins to ice-covered ridges, hydrothermal vents trace a global network along the ocean floor. Their distribution across the Pacific, Atlantic, Indian, Arctic, and Southern Oceans confirms that the deep sea's internal furnace operates worldwide. Even at the greatest depths yet explored, life gathers where heat and chemistry intersect, demonstrating that these hidden ecosystems are not isolated anomalies, but integral features of Earth's dynamic crust. Discovery did not end with seeing. Understanding required bringing fragments of this world into human laboratories, and that task proved far more difficult than descent itself. Many organisms adapted to crushing pressure and narrow chemical gradients cannot survive the journey upward. As a submersible ascends, pressure decreases steadily, altering the physical context in which every cell has evolved to function. Early expeditions relied on crude vehicles such as Alvin, whose mechanical arms collected samples into insulated baskets. These containers protected specimens from mechanical damage, but offered no defense against decompression. By the time the vehicle reached the surface, many animals had already perished. Tissues degraded quickly once removed from their native pressure and temperature, complicating physiological study. Remotely operated vehicles expanded access to deeper and more remote sites. Equipped with high-resolution cameras and dexterous manipulators, ROVs could remain submerged for extended periods, transmitting live video to researchers aboard ships. They allowed precise collection of organisms and fluids, reducing disturbance to fragile structures. Yet the fundamental challenge remained. Ascent changed the environment irreversibly for pressure-sensitive life. To address this problem, engineers developed pressure-retaining samplers. These specialized containers seal at depth, maintaining internal pressure during retrieval. Some are designed for fluids, capturing vent water without allowing dissolved gases to escape. Others hold small animals within chambers that preserve compression until laboratory analysis can occur in controlled facilities. Such devices function as portable fragments of the deep, carrying a piece of the abyss upward intact. Even with these innovations, sampling remains complex. Temperature shifts occur rapidly as equipment moves from warm venting zones into colder surrounding water, then into surface air. Chemical gradients dissipate once isolated from continuous flow. Symbiotic relationships may depend on conditions difficult to replicate outside the seafloor. Maintaining appropriate mixtures of dissolved compounds requires careful design of experimental systems. Laboratories capable of simulating deep-sea conditions employ high-pressure vessels and temperature control systems. Within these chambers, researchers can observe microbial growth or measure enzymatic activity under conditions approximating those in situ. Multicellular organisms are more challenging. Their larger size and integrated physiology demand stable gradients and fluid exchange that are difficult to reproduce precisely. Field studies, therefore, remain essential. In situ, experiments conducted directly on the seafloor have become more common. Instruments are deployed near vent structures to monitor temperature, chemistry, and biological responses over time. Some platforms allow small-scale manipulations, altering fluid flow or introducing substrates while recording changes. Such approaches reduce the need to transport sensitive organisms to the surface. Sampling also raises ethical and ecological considerations. Vent fields are limited and dynamic habitats. Excessive removal of organisms can disrupt local communities. As exploration expands, researchers strive to balance scientific inquiry with conservation, minimizing impact while gathering necessary data. The fragility of these systems extends beyond their response to geological change, encompassing vulnerability to human disturbance. Despite these obstacles, knowledge has grown steadily. High-pressure microscopy, molecular sequencing, and advanced imaging techniques reveal details of cellular adaptation and community structure. Pressure-retaining containers preserve microbes for genomic analysis, allowing insight into metabolic pathways and evolutionary relationships. Each technological advance narrows the gap between observation and understanding. Studying life that dies at the surface demands patience and ingenuity. It requires tools capable of crossing boundaries without dissolving the context that gives these organisms coherence. Through careful design and persistent exploration, scientists continue to illuminate systems that resist removal from their native depth. The challenge itself underscores how finely tuned these creatures are to pressure, temperature, and chemistry, reminding us that their survival depends on conditions rarely encountered beyond the seafloor. From the mineral pores and superheated fluids of the deep, molecules have traveled upward into laboratories and factories, carrying with them properties forged under extremes. The same adaptations that allow microbes to function at high temperature and pressure have proven unexpectedly valuable in human technology. What began as curiosity about life in darkness has unfolded into practical applications that reach far beyond the seafloor. One of the most influential discoveries concerns heat-stable enzymes. In most organisms, enzymes lose their structure and cease functioning when exposed to elevated temperatures. Hyperthermophilic microbes, however, produce proteins that remain active where others would unravel. Their catalytic sites retain shape despite vigorous molecular motion, allowing chemical reactions to proceed efficiently under conditions that denature typical proteins. These enzymes have been isolated, characterized, and reproduced through recombinant DNA techniques. Among the most transformative examples are thermostable DNA polymerases. In molecular biology, the polymerase chain reaction relies on repeated cycles of heating and cooling to amplify specific segments of DNA. Early versions of this method required replenishment of enzyme after each heating step, because conventional polymerases degraded. The introduction of thermostable polymerases eliminated that limitation, enabling rapid and automated amplification of genetic material. Although the most widely known thermostable polymerase originated from a terrestrial hot spring bacterium. Exploration of deep-sea vents has yielded additional variants with distinct properties. Some function optimally at different temperature ranges or possess proofreading capabilities that enhance fidelity during DNA replication. These differences expand the toolkit available for genetic research, diagnostics, and forensic analysis. Industrial processes also benefit from enzymes adapted to extreme conditions. Many chemical reactions proceed more rapidly at higher temperatures, reducing contamination risk and increasing efficiency. Heat-stable proteases, lipases, and amylases derived from thermophilic organisms can be incorporated into detergents, food processing, and biofuel production. Their resilience allows them to withstand harsh conditions, such as high salinity or variable pH, features common in industrial settings. Beyond enzymes, vent microbes produce novel biochemical compounds with potential medical relevance. Secondary metabolites synthesized in competitive microbial communities may exhibit antimicrobial or anti-cancer activity. Researchers screen extracts from vent-associated bacteria and archaea for molecules capable of inhibiting pathogens or modulating cellular pathways. While many candidates remain in early stages of investigation, the chemical diversity of these environments suggests untapped reservoirs of bioactive substances. Structural studies of heat-resistant proteins have also deepened understanding of protein folding and stability. By comparing thermophilic enzymes to their mesophilic counterparts, scientists identify features that enhance durability. This knowledge informs protein engineering, allowing the design of enzymes tailored for specific industrial or medical purposes. Modifications inspired by vent organisms can increase stability, extend shelf life, or improve performance under demanding conditions. The impact extends to evolutionary biology and biotechnology alike. Studying organisms that thrive in high-temperature systems clarifies fundamental principles of molecular stability, shedding light on how early life might have functioned. At the same time, practical applications translate those principles into tools that support modern medicine and manufacturing. The journey from vent to laboratory is not direct. It requires careful sampling, preservation of genetic material, sequencing, and expression in model organisms. Yet once genes encoding useful enzymes are identified, they can be synthesized and produced at scale without continued extraction from fragile habitats. In this way, the benefits derived from extremophiles need not compromise the ecosystems that inspired them. Within molecules shaped by geological heat, new possibilities have emerged. Enzymes that endure boiling temperatures enable rapid genetic analysis. Proteins, stable in harsh environments, support cleaner industrial reactions. Compounds forged in competitive microbial niches hint at therapeutic potential. From the deep sea's hidden chemistry, insights rise that influence medicine, industry, and our understanding of life's adaptability. Interest in the deep ocean no longer belongs solely to scientists. Beneath the quiet expanse of water lie mineral deposits enriched by hydrothermal circulation, concentrations of copper, zinc, gold, and rare elements that industry values. The same processes that build chimneys layer metals into sulfide mounds, creating ore bodies unlike those on land. As terrestrial resources grow harder to access, attention has turned toward these submerged formations. Exploratory surveys have mapped vent-associated deposits along several mid-ocean ridges and back-arc basins. Robotic systems sample crusts and sulfide blocks, analyzing metal content and thickness. The scale of some deposits suggests economic potential. Yet these mineral accumulations are not separate from the ecosystems described earlier. They are the substrate upon which communities assemble, the architecture that channels chemical gradients and supports attachment. Mining proposals typically involve remotely operated machinery capable of grinding and collecting sulfide material from the seafloor. Sediment plumes generated during such operations could disperse fine particles across surrounding habitats. Even limited disturbance might alter fluid flow pathways, changing the gradients that organisms depend upon. Because vent ecosystems occupy narrow bands of suitable conditions, small shifts can have disproportionate effects. Ecological fragility arises not from inherent weakness of individual species, but from the specialized nature of their habitat. Vent communities are localized, often confined to areas measured in tens or hundreds of meters. Many species have restricted distributions, found only along particular ridge segments. While larval dispersal connects populations over time, the distance between active sites can be vast relative to the scale of individual fields. Recovery from disturbance depends on several factors. If mining were to remove an active chimney and associated fauna, recolonization would require either continued venting nearby or reactivation of the site. Where hydrothermal activity persists, microbes might recolonize within months, forming new biofilms on exposed surfaces. Larger invertebrates would follow more slowly, their arrival contingent on larval supply and suitable chemical conditions. However, if extraction disrupts fluid circulation permanently, the energy source sustaining the community could cease. In that case, recovery would not occur until geological processes reopen pathways, a timescale measured in decades or longer. Even when activity resumes, species composition might differ from the original assemblage, influenced by chance arrival and altered microhabitats. Experimental disturbances have provided limited insight. In some cases where vent structures were artificially removed for study, microbial communities re-established relatively quickly once surfaces stabilized. Yet the return of complex assemblages required more time, and full restoration of prior diversity was not always observed within monitoring periods. These findings suggest resilience at the microbial level, coupled with vulnerability among specialized macrofauna. Sediment plumes pose additional uncertainty. Fine particles settling over adjacent habitats could smother filter feeders or interfere with larval settlement cues. The extent of plume dispersal in deep currents remains difficult to predict accurately. Modeling efforts attempt to estimate spread, yet real-world conditions vary with topography and flow patterns. International discussions now address regulation of deep-sea mining, balancing resource demand with conservation. Scientific assessments emphasize the uniqueness of vent ecosystems and the limited understanding of long-term impacts. Because many vent species remain undescribed, potential losses cannot be fully quantified. The prospect of mining introduces a new variable into a system already shaped by tectonic instability. Natural cycles of birth and collapse occur, yet they are driven by geological rhythms. Human intervention would operate on different timescales and spatial patterns. In a landscape where life organizes around precise intersections of chemistry and heat, disturbance carries consequences that extend beyond the removal of rock. Within these hidden oases, minerals and organisms are inseparable. Decisions about extraction will determine whether the deep sea's internal furnace remains solely a source of scientific insight or becomes a frontier of industrial expansion with outcomes still unfolding. Far from Earth's oceans, beyond the orbit of Mars, other seas may lie hidden in darkness. They are not open to the sky. They are sealed beneath kilometers of ice, locked inside distant moons that circle giant planets. Yet beneath those frozen shells, models suggest water remains liquid, warmed not by sunlight, but by forces that bend and knead their interiors. Europa, a moon of Jupiter, carries a surface etched with fractures and shifting plates of ice. Beneath that brittle crust, evidence points to a global ocean in contact with a rocky mantle. Enceladus, orbiting Saturn, reveals its secret more openly. From fractures near its south pole, plumes of water vapor and icy grains erupt into space, sampled by passing spacecraft. Within those jets, instruments have detected salts, simple organic compounds, and tiny silica particles consistent with hydrothermal interaction between water and warm rock. The source of heat in these worlds is tidal. As Jupiter and Saturn exert gravitational pull, their moons are stretched and compressed during each orbit. This constant flexing generates internal friction, converting orbital energy into warmth. Over time, that heating prevents complete freezing of subsurface oceans. In some regions, water may circulate through porous rock, much as seawater on Earth descends into fractured crust and returns enriched with dissolved minerals. The parallel is quiet but profound. On Earth, hydrothermal circulation creates chemical gradients that sustain ecosystems independent of sunlight. On Europa and Enceladus, tidal heating could drive similar circulation, producing interfaces where reduced compounds meet oxidized ocean water. If water interacts with silicate rock at elevated temperatures, hydrogen and other molecules can form through reactions between minerals and fluid. These chemical imbalances represent stored energy, the same kind of disequilibrium that vent microbes exploit. Astrobiology once centered largely on the search for planets within a narrow, habitable zone where starlight allows liquid water at the surface. The discovery of thriving communities powered by chemical energy reshaped that framework. Habitability expanded beyond distance from a star. It became linked instead to the presence of liquid water, essential elements, and sustained energy sources, regardless of illumination. Spacecraft observations have strengthened this shift. The detection of molecular hydrogen in Enceladus's plume suggests ongoing hydrothermal reactions within its interior ocean. Models of Europa's ocean floor predict pressures and temperatures compatible with water circulating through rocky crust. Although no probe has yet descended through their ice, the chemical fingerprints imply dynamic systems rather than static reservoirs. Life elsewhere would not need to mirror the forms found on Earth's seafloor. Even so, the existence of ecosystems sustained by chemistry alone provides a template. Microbial metabolisms that oxidize hydrogen, methane, or sulfur compounds could operate wherever such gradients persist. The absence of light would not be a barrier, as it is not a barrier in Earth's deepest waters. Future missions aim to investigate these possibilities more closely. Instruments designed to analyze plume material or map subsurface oceans will search for complex organic molecules and isotopic patterns suggestive of biological activity. The challenge lies in distinguishing chemical processes driven purely by geology from those influenced by metabolism. On Earth, that distinction often requires detailed sampling and contextual understanding. In contemplating oceans beneath ice, the narrative of life extends outward. The furnace within Earth's crust, once thought exceptional, becomes part of a broader category of planetary energy systems. Worlds previously dismissed as frozen and inert emerge as candidates for hidden seas warmed from within. The idea that life might exist in such remote environments does not rest on speculation alone. It rests on observation that biology can anchor itself to gradients of chemistry and heat. If similar gradients arise beneath distant ice, the possibility remains open that somewhere, in darkness far from any sun, water circulates over warm rock and chemistry edges toward complexity. If Earth were drained of its oceans, mountains would appear smaller than imagined, continents thinner. The planet's dominant feature revealed not as land, but as vast basins. The deep sea occupies the greater portion of that space. Below 200 meters, where sunlight fades to absence, an immense volume begins, extending down through plains, ridges, trenches, and vent fields. By sheer measure, this dark expanse constitutes the largest habitat on the planet. The shallow coastal margins that humans know intimately form only a narrow fringe. Even the sunlit surface waters represent a delicate skin compared to the depth beneath. More than 90% of the ocean's living space lies in darkness, under pressures and temperatures that shift gradually with depth but remain stable across immense distances. When viewed in terms of cubic kilometers rather than surface area, the dominance of the deep becomes clear. This habitat is not uniform. Abyssal plains stretch for thousands of kilometers, carpeted with fine sediment. Mid-ocean ridges rise like submerged mountain chains, their flanks fractured and porous. Trenches descend into Haddle zones where pressure climbs further still. Within this three-dimensional landscape, currents move slowly, redistributing heat and nutrients. The overall environment changes far less rapidly than climates at the surface. Over geological time, the deep ocean has offered continuity. While continents drift, glaciers advance and retreat, and atmospheric composition fluctuates, the abyssal realm remains buffered. Temperatures at depth vary within narrow ranges, insulated from seasonal cycles. Even during periods of planetary upheaval, such as mass extinctions triggered by asteroid impacts or volcanic outpourings, the deep sea may experience muted effects compared to surface ecosystems. Such stability allows lineages to persist across vast spans of time. Fossil records of deep dwelling groups suggest continuity stretching back hundreds of millions of years. Evolution proceeds there, but often at different rates, influenced by isolation and steady conditions. Some species display remarkable morphological conservatism, while others diversify in response to subtle ecological gradients invisible from above. Hidden biodiversity emerges from this interplay of space and time. Many deep-sea organisms remain undescribed, known only from occasional trawls or submersible observations. Soft-bodied invertebrates, microbial communities within sediments, and species inhabiting rocky outcrops contribute to a richness not easily quantified. Genetic studies frequently reveal cryptic diversity, multiple species concealed within what once appeared to be a single form. Energy sources in the deep vary. Beyond hydrothermal systems, organic material sinks from surface waters, a slow rain of detritus that nourishes benthic communities. In some regions, methane seeps and other chemical anomalies provide additional sustenance. The scarcity of food in many abyssal areas shapes life histories characterized by slow growth and long lifespans. Yet even there, adaptation finds pathways. Because the deep sea covers such a large fraction of Earth's biosphere, its influence on global processes is profound. It participates in carbon cycling, storing vast quantities of organic and inorganic carbon within sediments and water masses. Microbial activity at depth contributes to nutrient regeneration that ultimately supports productivity nearer the surface. The abyss is not isolated. It is integrated into planetary systems. Despite its scale, human awareness of this habitat remains partial. Exploration has sampled only a minute percentage of the seafloor. Each expedition tends to reveal forms and interactions previously unimagined, reinforcing the sense that much remains unseen. The deep sea is not merely an extension of familiar ecosystems into darkness. It is a dominant component of Earth's living space, vast, persistent, and intricately connected to the stability of the planet itself. In recognizing the deep as Earth's largest habitat, perspective shifts. What appears peripheral from a shoreline becomes central when viewed in volume and duration. Beneath the surface, in that expansive and enduring realm, life has unfolded across ages, largely beyond human notice, shaping and being shaped by a world defined more by depth than by light. In certain corners of the ocean, evolution moves to a different rhythm, where chemical flux replaces daylight and temperature gradients shift across the span of a hand, selective pressures sharpen. Heat, there is not a background condition, but an active sculptor, shaping proteins, partnerships, and life histories with persistent insistence. Thermal stress acts at the molecular scale. Proteins must fold into precise shapes to function, and elevated temperatures increase the tendency for those shapes to unravel. Within vent-influenced habitats, organisms that persist possess variants of enzymes stabilized by subtle changes in amino acid sequence. A single substitution can tighten internal bonds, reducing flexibility just enough to resist unfolding. Over generations, lineages accumulate these modifications, refining structures to match their thermal niche. Membranes also adjust. The lipids composing cellular boundaries determine fluidity, and temperature alters that fluidity continuously. Species exposed to warmer conditions incorporate lipid molecules that pack more tightly, preventing excessive permeability. In cooler adjacent zones, different compositions prevail. Because thermal gradients can shift over short distances, populations may experience microgeographic selection, favoring individuals whose cellular chemistry aligns precisely with local conditions. Adaptation in these environments is not solely genetic. Symbiosis emerges as a central strategy. Many vent animals rely on internal or external microbial partners capable of harnessing chemical energy. This alliance allows hosts to occupy habitats that would otherwise offer limited nutrition. The partnership is dynamic. Host physiology evolves to transport substrates such as hydrogen sulfide and oxygen efficiently, while symbionts refine metabolic pathways to convert those compounds into organic molecules. Heat influences these relationships as well. Symbiotic microbes must tolerate temperatures encountered within host tissues, and hosts must regulate internal conditions to maintain symbiont viability. In some species, vascular systems distribute chemicals while buffering thermal extremes, creating stable microenvironments despite external fluctuation. Evolution therefore operates across two genomes at once, selecting combinations that function coherently under stress. Rapid environmental change at vent sites further accelerates evolutionary processes. Hydrothermal activity can intensify or diminish over years to decades, altering temperature and chemical output. Populations exposed to such variability may retain genetic diversity that permits swift response. Larval dispersal between sites introduces additional variation, mixing alleles adapted to slightly different regimes. Over time, this interplay between isolation and connectivity generates mosaics of local specialization. Energy derived from chemical reactions expands ecological possibility. In habitats where sunlight never penetrates, metabolism based on oxidation of reduced compounds establishes primary production. Organisms evolving within this framework are freed from dependence on photosynthetic input, allowing colonization of deep settings long before complex surface ecosystems flourished. The existence of these pathways demonstrates that evolutionary innovation is not constrained to a single energy source. Heat, as an evolutionary force, extends beyond simple tolerance. It influences body size, growth rate, and reproductive timing. Some vent-associated species grow rapidly when conditions are favorable, capitalizing on abundant chemical energy. Others invest in longevity and delayed reproduction, strategies suited to more stable or resource-limited niches. These life history traits reflect integration of thermal and chemical factors over evolutionary time. Viewed across millions of years, the persistent presence of hydrothermal systems provides a recurring stage upon which adaptation unfolds. As tectonic plates shift and new vents form, lineages disperse, diversify, and occasionally converge on similar solutions. The patterns observed suggest that when energy gradients are strong and consistent, evolution explores predictable pathways. Within the deep ocean's hidden corridors, heat is both challenge and catalyst. It destabilizes molecules, yet drives reactions. It constrains physiology, yet enables new symbioses. Through countless generations, organisms have negotiated its demands, transforming thermal stress into opportunity. In doing so, they reveal evolution not as a slow drift alone, but as a responsive dialogue between chemistry, temperature, and the persistent capacity of life to adjust its design. Nothing at a hydrothermal vent is permanent. The chimneys that rise from the seafloor are constructed slowly by mineral precipitation, layer upon layer of sulfide crystals building fragile towers. Yet the same tectonic forces that create pathways for heated water also reshape the crust without warning. Over years or decades, a vent may weaken, fracture, or fall silent as fluid flow shifts elsewhere. Birth begins invisibly. Seawater seeps into newly cracked rock, descends, heats, and returns altered. At first, the discharge may be diffuse, shimmering across the seafloor. Minerals crystallize as the hot fluid meets colder surroundings, forming small mounds that grow steadily. Microbial films settle almost immediately, coating fresh surfaces with living chemistry. Within months, the first invertebrates may arrive, drawn by chemical cues that signal energy availability. Communities assemble in waves. Early colonizers often include fast-growing species capable of exploiting unstable conditions. Their larvae drift in from distant sites, settling where gradients are strong enough to sustain metabolism. As structures expand and flow stabilizes, additional species join, each occupying slightly different microhabitats along temperature and chemical contours. Over time, a layered ecosystem emerges, complex yet delicately balanced. Death can be abrupt. A minor seismic event may collapse a chimney, burying attached organisms beneath rubble. In other cases, magma intrusions alter subsurface plumbing, cutting off fluid supply. Without chemical energy, symbiotic relationships fail, and dependent animals perish or disperse. The site cools, leaving behind mineral remains and scattered shells. These cycles of emergence and disappearance are intrinsic to vent systems. Geological processes operate continuously along spreading ridges, generating new habitats even as older ones fade. For species inhabiting this landscape, persistence depends on mobility and timing. Many vent organisms release planktonic larvae capable of surviving weeks or months in the water column. During that interval, currents may carry them across considerable distances until they encounter suitable conditions. Resilience at the community level arises from this dispersal network. When one site is lost, nearby populations can seed newly formed vents. Genetic studies reveal connectivity among fields separated by tens or hundreds of kilometers, indicating repeated recolonization over evolutionary time. Yet connectivity is not uniform. Physical barriers and current patterns influence which populations exchange genes, shaping regional differences in species composition. Patterns of succession following disturbance provide insight into recovery dynamics. After a chimney collapse, microbial communities re-establish first, exploiting residual heat and chemical traces. Pioneer invertebrates follow, often species tolerant of broader temperature ranges. Gradually, as flow intensifies or stabilizes, more specialized organisms arrive. The sequence reflects both ecological interactions and physiological constraints. Despite frequent turnover, certain lineages have persisted along ridges for millions of years. Their continuity suggests that while individual sites are transient, the overall system maintains long-term stability at a broader scale. The vent field network functions as a shifting mosaic, with habitats blinking on and off, but never disappearing entirely from the planet. This interplay between destruction and return shapes evolutionary trajectories. Species adapted to rapid colonization may trade competitive dominance for dispersal efficiency. Others specialize in later successional stages, thriving only when structures mature. The result is a dynamic equilibrium sustained by geological renewal. In the darkness of the deep ocean, resilience does not imply resistance to change. It signifies capacity to reassemble after loss. Chimneys crumble, fluids reroute, communities vanish, yet elsewhere new plumes rise and fresh surfaces harden. Life responds not with permanence, but with movement and renewal, tracing the contours of Earth's restless crust and adapting to a world where stability exists only in the continuity of transformation. Long before any creature with eyes drifted through open water, before shells hardened or fins unfolded, the ocean floor was already restless. Along fractures in the young crust, heated water circulated through stone, rising and falling in cycles that predate continents in their present form. For billions of years, this exchange between rock and sea has continued, largely unseen, shaping chemistry in the dark. The deep ocean measures time differently from the surface. Storms do not churn its depths, seasons do not alter its temperature in rapid swings. Sediments settle slowly, grain by grain, recording ages in thin layers. Within that steady environment, hydrothermal systems have flickered on and off across geological eras, each vent field a temporary expression of a process that endures far longer than any individual structure. Evidence from ancient rocks suggests that similar circulation may have occurred on the early Earth, when the planet's crust was thinner and internal heat greater. Chemical signatures preserved in some of the oldest formations hint at interactions between seawater and hot basalt. Though direct traces of early life are rare, isotopic patterns imply biological activity dating back more than 3 billion years. In that immense span, countless vent systems would have formed and faded, sustaining microbial communities beyond the reach of light. Most of this history unfolded without observation. Even in recent centuries, the deep sea remained inaccessible, its processes inferred rather than witnessed. While human societies rose and fell on land, and surface ecosystems shifted with climate and evolution, the abyss maintained its quiet cycles. Vent communities assembled, dispersed, and reassembled again, unnoticed except by the organisms that depended on them. The scale of deep ocean time challenges ordinary perspective. A vent may remain active for decades or centuries, yet the tectonic setting that produces such vents persists for millions of years along spreading ridges. Over that duration, thousands of individual sites may ignite and extinguish, their cumulative activity forming a continuous thread of chemical exchange between Earth's interior and its oceans. Life adapted to this rhythm carries within it echoes of that antiquity. Microbial lineages inhabiting hydrothermal environments often display deep evolutionary roots, branching early in the tree of life. Their metabolic pathways, tuned to exploit inorganic compounds, may resemble strategies employed on the young planet. In that sense, modern vent ecosystems offer glimpses into processes that have operated since near the dawn of biology. Yet the persistence described here is not static repetition. Each geological epoch introduces variation in ocean chemistry, atmospheric composition, and tectonic configuration. Vent fluids interacting with differing crustal materials generate distinct chemical mixtures. Organisms respond, diversifying or declining according to new conditions. Over immense spans, adaptation and extinction weave together, maintaining continuity through change. The unseen nature of this persistence underscores its scale. Even today, vast portions of the seafloor remain unmapped in detail. Entire vent fields may operate beyond the reach of current exploration, their communities cycling through growth and collapse without documentation. The ocean's depth acts as both cradle and curtain, sustaining processes while concealing them from casual view. To contemplate billions of years of hydrothermal activity is to recognize a continuity that dwarfs human timelines. Civilizations occupy moments measured in centuries. Vent systems participate in a planetary narrative measured in eons. Through shifting continents and evolving atmospheres, the exchange of heat and water at the seafloor has remained a constant. Supporting life informs both ancient and newly emerged. Within that enduring framework, the quiet persistence of the unseen becomes part of Earth's identity. Beneath waves that reflect sunlight and weather, a deeper chronicle unfolds, marked not by seasons but by tectonic drift and chemical flux. Life has accompanied that chronicle for most of the planet's history, adapting, dispersing, and returning in patterns that extend far beyond immediate perception. Sustained by the steady dialogue between ocean and stone, limits are often drawn before they are tested. For much of scientific history, boundaries for life were defined by familiar experience, moderate temperatures, breathable air, sunlight as the primary source of energy. Beyond those conditions, existence was considered improbable. Yet in the depths we have explored, organisms persist in places once categorized as impossible, not by defying physics, but by inhabiting its margins with precision. Habitability, once equated with warmth and light, now extends into realms shaped by pressure, chemistry, and heat rising from within a planet. The presence of liquid water remains essential, but the source of energy sustaining metabolism need not originate from a star. Chemical disequilibria, maintained by geological processes, can provide steady gradients for life to exploit. In that recognition, the concept of a habitable environment broadens beyond narrow climatic bands. These creatures reveal that biological boundaries are not fixed lines, but dynamic frontiers. Proteins that remain stable at temperatures once thought destructive demonstrate that molecular architecture can shift through incremental change. Membranes resistant to crushing pressures show that cellular structures adapt through subtle adjustments in composition. Symbioses that convert toxic compounds into nourishment illustrate that constraints may become resources under the right evolutionary pathways. Expansion of limits does not imply that life can thrive anywhere. Each organism occupies a specific niche defined by temperature, pressure, and chemical availability. Survival at extremes requires alignment between physiology and environment at fine scales. However, the range of viable combinations is wider than earlier models suggested. The discovery of thriving ecosystems in scalding, acidic, metal-rich settings underscores that biological potential extends into conditions once dismissed as sterile. This shift in understanding reframes Earth itself. From a surface perspective, our planet appears governed by sunlight and atmospheric dynamics. Yet beneath the waves lies a domain where internal heat drives ecosystems independent of the day-night cycle. In those depths, landscapes resemble alien terrains more than familiar seascapes. Mineral towers, venting superheated fluid into near-freezing water, create juxtapositions that challenge intuition. Recognizing Earth as more complex than assumed invites humility. What seems ordinary at the surface represents only a fraction of planetary diversity. The deep sea demonstrates that vast habitats operate under rules distinct from those shaping forests and grasslands. When considering other worlds, it becomes clear that our reference point has been incomplete. These organisms also teach that resilience often arises from cooperation. Symbiotic partnerships expand metabolic possibilities, allowing hosts and microbes together to occupy niches inaccessible alone. Such alliances suggest that limits may be crossed not by solitary adaptation, but through integrated systems that distribute functions across multiple partners. In contemplating limits, one encounters scale. At the microscopic level, enzymes adjust through minute changes in bonding patterns. At the ecological level, communities assemble where gradients persist. At the planetary level, tectonic and chemical cycles sustain energy flow over billions of years. Boundaries emerge differently at each scale, yet at all levels, life negotiates with its surroundings rather than passively enduring them. The creatures of the deep do not erase constraints. Instead, they redraw the map of possibility. They show that life can anchor itself beside boiling water under immense pressure, provided that microenvironments align and chemistry cooperates. They reveal that assumptions rooted in surface experience may overlook vast domains of viability. Through their existence, our understanding of limits evolves. Habitability becomes a spectrum shaped by energy sources and environmental stability. Biological boundaries expand as evidence accumulates. Earth, once perceived as comfortably familiar, reveals aspects that appear almost extraterrestrial when viewed from the seafloor upward. In acknowledging these lessons, the notion of what is extreme shifts. The extraordinary becomes part of a broader continuum, reminding us that the planet holds capacities beyond initial expectation. Limits remain, yet they are discovered not through speculation, but through exploration and the patient unveiling of life where it was least anticipated. At the farthest reach of lightless water, where the crust of the planet thins and fractures, heat rises without flame. It does not flicker like fire in air. It moves through stone and sea in slow circulation, invisible until it emerges as a plume against the cold. From that meeting of extremes, a paradox unfolds, one that has guided this descent from the beginning. Water heated beyond the boiling point, known at the surface, flows as liquid under immense pressure. Around it, the surrounding ocean remains near freezing. Within centimeters, gradients sharpen from scalding to tolerable, from lethal to sustaining. In that narrow band, life anchors itself. The contradiction is not resolved by denying physics, but by inhabiting its edges with precision. Earth's interior is not silent. Residual heat from planetary formation and the steady decay of radioactive elements within the mantle drive convection deep below the crust. Plates drift, separate, and collide. Along spreading ridges, magma approaches the seafloor, transferring energy into circulating seawater. The result is a network of hydrothermal systems that lace the ocean basins, each one a localized expression of a planetary engine. That internal power operates independent of sunlight. While surface ecosystems pulse with the rhythm of day and season, the deep relies on chemical exchange between rock and water. Reduced compounds carried upward become fuel for metabolism. In this exchange, geology and biology intertwine. The planet provides gradients, organisms refine pathways to use them. Life thriving in such conditions does not negate fragility. It persists by aligning with specific combinations of temperature, chemistry, and pressure. Too close to the hottest flow, proteins destabilize. Too far from chemical input, energy dwindles. Survival depends on occupying the precise intersections where extremes soften into viability. The existence of these intersections challenges assumptions about where living systems can function. Throughout this exploration, patterns have emerged. Communities assemble where heat and chemistry meet. They disperse when vents fall silent. They return when new pathways open. Microbes, invertebrates, and predators participate in cycles shaped by tectonics and time. Each species reflects adaptation to gradients that would seem hostile from a distance. The heat beneath the sea reminds us that Earth is powered not only from above but from within. Solar radiation drives winds and currents, yet internal energy shapes continents and fuels hidden ecosystems. Together, these forces create a planet layered with environments that differ profoundly from one another, each sustained by distinct sources. In reflecting on boiling water and survival, the paradox becomes less a contradiction and more an illustration of balance. Extremes coexist, separated by scales so fine they can be crossed by the span of a limb. Life occupies those scales, adjusting molecular structures, forging symbioses, and dispersing across vast basins to persist. The ocean floor remains largely unexplored, its furnace still active, its gradients still forming new habitats beyond observation. Even with decades of study, each descent reveals structures and organisms that refine understanding further. The narrative does not close with certainty, but with an expanded sense of possibility grounded in evidence. Beneath the waves, where heat rises through fractures in stone, the planet continues its quiet exchange of energy and water. Within that exchange, life has found a way to endure, not in defiance of extremes, but in dialogue with them. The sea above may appear calm, yet below it, an internal warmth sustains communities shaped by contradiction, reminding us that the boundaries of the living world are drawn by processes far deeper than the horizon reveals.